This program is brought to you by Stanford University. Please visit us at stanford.edu.
Well, Irv, coming from you, that is high praise indeed. Irv is the guy who catches the most fish on all these trips we take together, and I resent it. I want to begin by thanking the Tseng family and their friends for establishing this lectureship and then inviting me to speak in it. These events, which focus on individuals who have learned about the dreadful aspects of cancer, are important. This occasion brings to mind an occasion 14 years ago when about 150 of us assembled at the Fred Hutchinson Cancer Center to talk about glioblastoma, which was then afflicting our friend Hal Weintraub, who died soon thereafter. And I think many of us at that gathering, exploring what we knew and didn't know, especially what we didn't know about glioblastoma, came away from that event inspired to do something about it, realizing that the tools we now have in hand were powerful enough to teach us things we didn't know and hopefully would make the disease a less dreadful one. My own research career was affected by that. Went home to NIH and began to use viral delivery systems to deliver cancer genes to glial cells. And Eric Holland, who was actually a graduate of this medical school, took up that work and is now the leader of the Brain Tumor Center at Sloan-Kettering Institute. And I think that just illustrates how one thing leads to another, and I think emulates or exemplifies the kind of effect that lectureships and occasions like this can have. Now, I realize this is a diverse audience, and my intention today is to try to provide some entertainment and information for everybody. I'll try to make the language plain. I am going to talk about this cultural phenomenon of bringing the study of the biology of cancer, the basic science of cancer, together with the clinical aspects of cancer and doctors who take care of patients who have the disease. But I also want to tell about some science that we're doing in my lab, and I'll try to do that in as clear a fashion as possible without boring my friends who are basic scientists here at Stanford. Let me begin by a brief historical reflection. Irv was kind to say that Mike and I began doing our work in the late 70s. The truth is we began working together around 1970, a longer time ago than any of us like to contemplate. And at the time that we began working at UCSF, it did seem that there were two spheres of activity at that center. One sphere of people who were interested in how cancer began and others who were taking care of cancers in the clinic. These two groups of people were like two civilizations, different cultures, different languages, different times. They came to work at different times, rarely spoke intelligently or amiably to each other, like two spheres of religion and science. One of the gratifying things about what's happened in the last several years, last 15 or 18 years, is that these two worlds are beginning to overlap. And that's true for a number of reasons that we'll be exploring. Among the manifestations of that overlapping, introduction of antibodies like Herceptin that are active against oncogenic proteins are now known to be effective in reducing the incidence of breast metastasis, for example. The beginnings of efforts to assess individuals for risk of cancer by looking at inherited mutations that affect genes that we know are involved in carcinogenic processes. The use of gene expression patterns and efforts to define mutations present in cancer for making more precise diagnoses, for making assessments of a patient's likelihood of survival and response to therapies that are targeted against some of the damage we know exists in cancer cells. And then we have a few drugs that actually inhibit enzymes that are encoded by oncogenes, and I'll be talking about a few of those today. Now, this has been possible because of two and maybe three broad areas of discovery that have affected the way all of us think about cancer. First is the notion that cancer arises from mutation. The cancer is basically a genetic disease, largely one of somatic mutations, but inherited mutations play their role as well. And these changes, which are generally multiple, affect three classes of normal genes. One, the class we call proto-oncogenes, which when they've undergone mutation, act in a dominant fashion. Tumor suppressor genes, which play a recessive role in tumorigenesis, and genes that govern DNA integrity and contribute to cancer because loss of DNA repair functions lead to mutations in the other two classes of genes that more directly participate in carcinogenesis. The second major area of research is to understand what those genes do, what kind of proteins they make, where are the proteins situated in the cells, what are their physiological functions. And important among those functions for today's discussion will be the fact that many of these genes encode, many of the oncogenes in particular, encode enzymes, particularly protein tyrosine kinases, that will be the focus of some of our discussion as we go along today. The third big development that will be most important in thinking about some of the experiments I'll tell you today is that we've learned over the last several years that oncogenes not only maintain a cancerous state, but they are required to maintain the viability of cancer cells. This leads to the idea that cancer cells are dependent on the continued expression of the oncogenes that have initiated the growth of those cells, or more colorfully, perhaps less accurately, cells are addicted to continued expression of the oncogenes. And this obviously makes proteins encoded by oncogenes to be particularly attractive targets for therapeutic intervention. Now, obviously, pulling this information together leads us to a brief moment of inspiration here about a world in which clinical oncology and molecular understanding of cancer cells creates one world in which everything we do as physicians taking care of cancer patients, whether it's assessing risk or making diagnosis or trying to prevent or treat cancer, is based on a detailed understanding of how that happens. We're not there yet, but one of my objectives today is give you some idea of how I think we'll get there. So let me then step back from history and tell you what I think is going on in this field in very broad terms, then give you a couple of anecdotes from my own lab that illustrate how we're approaching some of these new themes. Now, the first obvious missing ingredient in our understanding of cancer is knowing everything that leads to the development of a cancer cell with its full panoply of characteristics. That is, its ability to grow when it should not grow, to invade, to metastasize, to recruit blood vessels, to escape cell death signals. And to do that, we need a clearer and deeper understanding of the mutations that affect genes in cancer cells, epigenetic changes that lead to alterations in gene expression. That's something which is now in motion as a result of a number of projects, some sponsored by the NIH, some not, and I'll mention a few of those as we go along. The second has to do with focusing not just on the primary tumor cells themselves, but on progression of tumors to form metastasis, which in the case of tumors outside the brain is the aspect of cancer that it kills most patients. Understanding more about the microenvironment, the stroma, the immune cells that respond to cancers. The third thing is to try to change our approach to therapy, more targeted therapy using antibodies and small molecules and possibly inhibitory RNAs that target the aspects of a cancer that are relevant to that particular cancer. And then once one has defined new targets and new drugs, dealing with an inevitable problem of drug therapy as it exists today, and that is understanding and overcoming drug resistance. And I will spend some time on that topic when I present one of my two anecdotes. And then a third theme that is particularly pertinent to today's talk is how we exploit mouse models, not just to validate the importance of all these genes I've been talking about that play a role in cancer, but to actually explore some of the questions about tumor progression and tumor therapy that are crucial to improving cancer care. One thing that's not on my list is the phrase I've heard most commonly today, cancer stem cells. That doesn't mean it's not important, but it's the last time I will use that phrase today unless I'm asked a question. Sorry, Irv. I think my qualities as a fisherman have just deteriorated a little bit, but you have more to say about than I do. So what I'd like to do today is tell you two stories about models and that address therapeutic issues. That is how we are attempting to use mouse models to think about targeted therapy of important cancers. And the first story, basically what I'm going to try to do for you is to tell you how we've tried to approach the question of how we cope with the problem that most cancers have multiple genetic abnormalities. So how do we selectively and successfully treat cancers in which the genotype is complex? And we're going to do that in the case I'll present by using multiple transgenes, two, one of which is regulated and one of which is not, and show you a collaborative model that is a model of breast oncogenesis in which two genes work together to make cancer, and I will show you that one oncogene is more important than another, and attacking two is still more important. The second anecdote, I'm going to tell you a story about lung cancer, lung adenocarcinoma, in which we are attempting to understand why patients who respond effectively to anti-tumor therapy that's based on inhibition of tyrosine kinases become drug resistant. And I'm going to show you how mouse models can be used to recapitulate the resistance that's seen, and in some cases actually first defined in human cancers. So the first story is a story that involves work done by Katrina Potsapanina, a woman who was trained as an MD in Russia, got her PhD at Columbia University, and then has been in my lab as a postdoc for several years studying breast cancer. And what I'm going to tell you is how she's made transgenic mouse lines that independently regulate the oncogene MYC or the common oncogene K-ras. For those of you unfamiliar with these terms, MYC encodes a transcription factor. K-ras encodes a GTP binding protein. Both of these genes are very, very commonly implicated in human cancer, but we don't have effective therapies that interdict the actions of either of these genes' protein products. We're going to use those genes in a cooperative fashion to induce tumors, and I'll show you that they do cooperate. Then I'll show you that the de-induction of either one or both causes tumor regression. The tumors are dependent on continued expression of these oncogenes. And then I'll show you that despite tumor regression, tumors recur. And by looking at the time it takes for tumors to recur, we get some sense of which of these targets is most important. In a sense, for those of you who are trying to get a handle on what I'm getting at, de-induction by using a regulatory system is like having an effective drug. We don't have drugs that work against MYC protein and RAS proteins, but by using transgenic systems in which the genes are regulated, in effect, we're interfering with one gene or the other by turning the gene off in the absence of a drug. And then I'll tell you about a little bit about why these tumors recur. Even when it seems that the tumor has completely disappeared, these animals are not cured, and tumors do recur, and in most cases, the tumor recurs because a mutation has occurred in a regulator of gene expression, and I'll explain why that's significant in a moment. So before I get to this story with mouse models, I need to tell you something more about oncogene addiction or oncogene dependence. And the best way to do this is to remind you that we have, as a result of some extraordinary work by Brian Drucker and many others, a poster child for oncogene dependence in the actual treatment of human cancer. And that poster child is a drug called Imatinib, or commercially Gleevec, a drug which inhibits at least 3 protein tyrosine kinases, one of which is the Abl tyrosine kinase, a kinase which is activated by a chromosomal translocation that creates the BCR-ABL protein, a constitutively activated protein tyrosine kinase, a protein tyrosine kinase that is characteristic in its activated form of the disease chronic myeloid leukemia. And patients who receive this drug almost always go into a very profound remission. There are now, in addition to patients who respond to this drug, patients with at least 5 other diseases, including one solid tumor, the gastrointestinal stromal tumor, or GIST, all of which are dependent on oncogenes that encode proteins that are inhibited by Imatinib. All of these diseases are treatable with Gleevec. The one serious shortcoming, aside from the expense of this drug, is that drug resistance occurs at a rate of about 1% to 4% a year in the case of CML, and it's usually due, almost always due to mutations in the kinase domain of ABL. And that one such mutation is the mutation I'll be referring to again later that results from a change of a threonine to an isoleucine at a particular position, but I will refer to that briefly a little bit later on. Now, the work with Imatinib has been a very powerful force in our field because it shows that trying to study oncogene dependence is obviously an exercise with incredible importance in treating human cancer. But many other diseases caused by other oncogenes have been much more difficult to approach. Another way to begin to both document and, more importantly, perhaps to study oncogene dependence is to make use of mouse models, and particularly mouse models in which oncogenes are under a simple form of transcriptional regulation. And that regulation is usually based on a system that was imported from bacterial studies some years ago that makes use of the antibiotics tetracycline and doxycycline. And the basic idea here, before I show you some details, is that when you induce the oncogene, tumors occur, and you de-induce it, and cancer cells die by the process known as programmed cell death or apoptosis. Now, just to give you a brief idea for those of you unfamiliar with this, I expect many of you are perfectly familiar with it, the idea here is that you set up a system in which an oncogene is under the control of an operator element, a promoter operator element that is responsive to a regulator from bacteria. And that regulator can either can respond positively or negatively to tetracycline or doxycycline. In the case of experiments I will describe today, the addition of the antibiotic turns these genes on. So in this case, there's a tetracycline activator called RTTA that is expressed by another transgene that has a tissue-specific promoter. In the case of the first experiments I'll describe, that promoter works in the lung in type 2 pneumocytes. Sorry, the first experiments I'll talk about, the promoter works in the mammary gland under the control of the mammary tumor virus long terminal repeat. In the second class of experiments, the promoter element for RTTA will be under the control of a lung pneumocyte-specific promoter. And the basic idea is that when you deprive the animal of doxycycline, the oncogene is off. When you add the antibiotic, the oncogene is turned on. If you remove the antibiotic from the diet, then the oncogene goes off again. Now, many genes have been implicated as oncogene dependence factors in mouse models by this route. And one of the first and most important demonstrations of the utility of this approach comes from a colleague of yours, Dean Felcher, who, when he was a postdoc with my colleague Mike Bishop, showed that c-Myc would induce a variety of hematological tumors in mice. And when c-Myc oncogene was de-induced by manipulation of doxycycline, that these tumors very rapidly disappeared. And this kind of result has been seen in a number of contexts, in melanomas using a RAS oncogene, in B-cell tumors using BCL-ABL, in hepatomas using the MET oncogene, and a variety of mammary tumors in Chodish's lab. I'm going to give you yet another example, because it serves my purposes later on, an example of oncogene dependence in which the K-ras oncogene, a common oncogene we're going to talk a little bit more about in a moment, was used to induce lung adenocarcinomas. And just to give you a sense of how these experiments are done, here you take one mouse that has a tetracycline regulator, RTTA, under the control of a lung-specific promoter, and a K-ras oncogene that has a mutation at codon 12, for those of you who are familiar with this idea. And you cross these two animals to make bi-transgenic animals. that will express mutant K-ras in the lung when given the tetracycline analog. And just a simple demonstration of what the results look like. Here is a piece of normal lung from an animal that has not received doxycycline. The lung looks perfectly normal. If you add doxycycline to the diet within a few months, you see many areas of adenoma and adenocarcinoma in these animals. And within days after removing doxycycline from the diet, the lung looks perfectly normal. I would also point out to the cognoscenti in the audience that this experiment is done in an animal that's also deficient for a tumor suppressor gene. So you don't need to correct everything in this tumor cell to allow oncogene dependence to exert its powerful force. The cells in this lung are still p53 deficient, but they are very dependent on sustained expression of a mutant K-ras gene. Now, how does this happen? Well, unfortunately, we don't really understand it, but there is a big idea behind all this that gives all of us something to think about. After all, there was a time when these lung cells were perfectly happy without any mutant K-ras. You induce expression, the cells become oncogenic. They, of course, are still viable. You turn off the oncogene, and now the cells die. Well, why does that happen? There's much debate in the field about why it happens. This is one of the biggest unsolved problems in cancer biology, in my view, and learning about the pathways that are affected by turning on an oncogene to make it so dependent on the oncogene product seems to me extraordinarily important. Now, in thinking about all these experimental models in which we turn one oncogene on, get a tumor, turn it off, and the tumor goes away, there have always been critics who say, well, that may work in these mouse models. Maybe it works in human CML, which seems to be dominated by the BCR-ABL fusion protein. But most human cancers probably have lots of mutations, not just two, probably more than that. Maybe this notion of oncogene dependence is not going to be so useful. And in any case, we're not going to have targeted drugs against all these oncogenic proteins. What are going to be the most useful for therapy? Is it necessary to target all of the mutant proteins, or can we achieve something by just targeting a single gene? So to approach this, as I said in my remarks a few minutes ago about Katrina Pozzopanina's experiment, we made models with independently regulated oncogenes that cooperate in tumorigenesis, and in order to test oncogene dependence and tumor recurrence in that setting. So what Katrina did was to both gather some mouse lines from some of our friends and make some of our own, and put together a set of mice that I'll now ask you to try to keep track of, mice in which both MYC and K-ras are DOX-inducible. All these lines, of course, can be bred to make tri-transgenic animals in which there's also a TET regulator, RTTA, being expressed. Second, we have mice that have MYC expressed constitutively, K-ras is inducible. And finally, the inverse, in which MYC is regulatable, K-ras is constitutively expressed. I remind you that the notion that these two genes might work together to make tumors is not an entirely novel one. Indeed, 20 years ago, Phil Leader did an experiment in mice in which he took two constitutively expressed genes, a Harvey RAS, similar to K-ras, but somewhat different, and a MYC gene, again constitutively expressed, and showed those cooperate to make tumors. But, and I remind you that these are very common human oncogenes for which interventions do not exist. Therefore, we have to use regulation to simulate therapy. So there are basically 3 or 4 kinds of questions we need to ask once we've made these mice. First, do they really collaborate, as old evidence would suggest? But do these animals in particular, with inducible oncogenes, actually collaborate in tumorigenesis? And I'll show you that in a moment. The answer is yes. Secondly, is one oncogene or are 2 required to maintain the tumors? And let's test that by looking at tumor regression after de-inducing one or both of the oncogenes. And then thirdly, is this kind of treatment more effective when we are de-inducing just one, either one, or both of the oncogenes? And to do that, we're going to wait and observe for tumor recurrence and then try to understand why recurrence has occurred. So first, this slide simply demonstrates that these genes do collaborate. So here, these are Kaplan-Meier plots in which on the y-axis we show animals without tumors and time on doxycycline along the x-axis. And here you see the difference between expressing just a single inducible oncogene, the MYC oncogene, as opposed to expressing both MYC and RAS under the control of the regulator. And you can see a rapid acceleration in the appearance of tumors. And similarly, by inducing MYC in the presence of constitutive RAS or inducing RAS in the presence of constitutive MYC, it's a dramatic acceleration of tumorigenesis. Not equal in all cases, but very convincing. Well, how does this look? Because the tumorigenesis is extremely rapid in this setting. And in fact, if you look at whole mounts of mammary glands from tri-transgenic animals, you can see that even after a single week on doxycycline, there's hyperplasia distributed throughout the mammary gland. By 2 weeks, this is nearly confluent, and you begin to see multifocal tumors within 5 weeks on doxycycline. Very rapid tumorigenesis indeed. If cells from these fat pads are injected intravenously, they go to the lung and make tumors at high frequency. Now, if we de-induce both of these oncogenes, both MYC and RAS, tumors disappear very rapidly. Within 2 days, there's already a lot of clearing of the fat pad. By 2 months, the mammary tree looks more or less normal compared to a non-transgenic. We also see tumor regression, dramatic tumor regression, if we turn off only the RAS oncogene. These are slices of mammary gland, and you can see that by 7 days after de-induction of RAS, there is much less cellularity. By 2 months after removal of doxycycline, the glands look essentially normal. When MYC is de-induced, we see an obvious gross effect. Tumors don't disappear entirely. But here's an animal with many, many large tumors before dox is withdrawn. We take the mice off doxycycline and de-induce the MYC gene. Within 7 days, there's a dramatic decrease in the amount of tumor. It's hard to see. This picture is not very well taken, but after a month, small, palpable tumors, but markedly reduced in size from the original tumors are evident. When we try to follow these tumors by looking, for example, at rates of apoptosis, demonstrated here by caspase-3 staining, you can see that all cases, whether when both genes are de-induced, when one, just the MYC gene is de-induced, or just the RAS gene is de-induced, dramatic increase in apoptotic cells occurs within a day or two after de-induction. And in most cases, we see a dramatic reduction in replication rates as well. Now, it's clear that from those experiments that the animals develop tumors in a cooperative fashion when both MYC and RAS are expressed, that either MYC or RAS alone, but more dramatically with the induction of RAS, or with the induction of both, tumors disappear. Well, do they reappear? Well, yes, they do. And if we look at animals that have recurrence of tumor after withdrawal of doxycycline with both MYC and RAS under dox control, we see tumors appearing in about half of the animals within a year of life. The same animals never on doxycycline don't have tumors at all. So that tells you that there's something about the experience of having a tumor that makes it more likely for a tumor to recur. There presumably were tumor cells in this population, in these animals that have the ability to grow into tumors again, and we'll ask in a moment why that's so. If we do the same experiment by de-inducing RAS, again, tumors do recur in these animals. They occur more frequently than in animals that have a constitutively expressed MYC oncogene, but don't express RAS because the RAS gene is under tetracycline control, and these animals are not seeing doxycycline. So there is recurrence actually is a more frequent event than the appearance of a primary tumor in animals that have a constitutively expressed MYC gene. And this converse is true for animals in which MYC was de-induced and have a constitutively expressed RAS gene. Again, recurrence occurs in nearly all animals, and it occurs earlier than the appearance of tumors in animals that express the RAS gene but not the MYC gene. So if you put all these together, and I know this is a fair amount of information, but if we put this all together, there's clearly some implications for therapeutic strategies. If we had drugs that interfered with MYC and RAS, and we were treating tumors that had human tumors that had RAS and MYC activated, we would have some basis for making some decisions. And that's perhaps best looked at by summarizing the data in the following way to ask, how long does it take for recurrence to manifest itself if we effectively treat the MYC oncogene, effectively treat the RAS oncogene, or treat them both together. And what this graph shows you is that, that MYC therapy has its benefits, but it extends life only by a few weeks, whereas treating RAS by turning it off allows the mouse to live another 3 months or so, and by knocking out both genes, by de-inducing them, we provide another 6 months or so of life. So one can develop a hierarchy of therapy based on this kind of result. Now, some of you may be asking, why did these tumors recur? And there are some obvious possibilities. One would be that the DOX-inducible oncogenes were somehow turned on again by a reactivation event that we can only make guesses about. Secondly, there might be secondary mutations in other cancer genes and residual cells that weren't palpable, or there might be epigenetic changes. And some recent results from Katrina suggest that the first of these is true, that the oncogenes are reinduced. That was found by doing reverse transcriptase PCR to show that in the animals that have an inducible MYC gene, MYC gets turned back on in the absence of doxycycline in about a third of the tumors that recur. In animals that have an inducible RAS gene, or both RAS and MYC, we see that the RAS gene in this case, both the RAS and the MYC gene in these cases are back on. Well, why might that have happened? First of all, it tells us something quite interesting, that it's more likely that a tumor will recur because you reactivate the pre-existing oncogene than by having mutations in other oncogenes, which we know have the potential to make mammary tumors out of mammary epithelial cells. So it suggests that the cells are particularly susceptible to the effects of these particular oncogenes. But what we found was that when we sequenced the RTTA tetracycline regulator, that in virtually every case we found, or she found, mutations that changed the nature of the regulator. And we were able to show that these regulators are now able to turn on teto oncogenes in the absence of tetracycline. So it basically changed the regulator so that it is now tetracycline or doxycycline independent. And in a sense, this doxycycline independence is like therapeutic resistance. In this case, therapy is taking away doxycycline, turning the gene off. And these cells become cancer cells again because they undergo a mutation that basically overcomes this, quote, therapy and result in resistance to the therapy of doxycycline removal. I think that's worth bearing in mind for the story I'll tell you next. So just to summarize what I told you before, what Katrina was able to do here to explore this notion of how you might model therapies that are pertinent to tumors that are known to be genetically complex is to independently regulate cooperating oncogenes MYC and RAS, to de-induce them independently or together, and to show by looking at tumor recurrence patterns that MYC seems to be a less useful target than RAS, and RAS alone a less useful therapeutic strategy than inhibiting MYC and RAS together. This is something that a general strategy that could be applied to other oncogenes and ought to be, and applied to other oncogenes and other tissue settings. As I illustrated at the end, when recurrence does occur, it's often appears to be the result of mutations that affect the regulator, suggesting that cancers are more likely to arise by reconstituting the genotype that produced the tumor in the first place, rather than by creating some additional genotype, not unlike what happens in CML that becomes resistant to ABL inhibitors, that is, ABL mutations that confer resistance to the effects of the ABL inhibitor, Imatinib, seem to be more frequent than bypass mutations. Okay, so let me move to the other story, which is a little simpler and will take less time, but equally interesting, I think. And that's to tell you about our efforts to understand drug resistance in a model of human lung cancer created in the mouse by using mutant forms of epidermal growth factor receptor. What I'm going to tell you in brief is something about human adenocarcinoma of the lung, and especially about that sector of human adenocarcinoma of the lung that has EGF receptor mutations that generate drug sensitivity in those tumors. And I'm going to show you a mouse model, or a set of mouse models, in which doxycycline-inducible mutant EGFR induces those cancers, and they're clearly EGF receptor dependent, just as they seem to be in humans. And what I will then show you is that we can generate drug-resistant tumors by manipulating the dosing of these animals with a tyrosine kinase inhibitor called erlotinib. And I'll show you that the mutations that confer resistance are the same ones that we see in human patients, namely a specific mutation in the EGF receptor at position 790, and amplification of another transmembrane tyrosine kinase gene, the MET gene. So a few reminders about human adenocarcinoma and what we've learned about them in the last few years. We know that, first of all, lung adenocarcinoma in human beings is one of the most common cancers, certainly one of the most common lethal cancers. There are about 1 million cases of lung cancer worldwide, 165,000 cases in the US, and a number of deaths that's almost equal to that. Roughly just under half of those tumors are adenocarcinomas, and of the adenocarcinomas, about a third, 20% to a third, have mutations in K-ras, the gene we were talking about in the context of breast cancer models a moment ago. About 10% have mutations in the epidermal growth factor receptor. Others, smaller numbers, have mutations in other EGF receptor pathways, PI3 kinase, the catalytic domain, a gene called BRAF that makes a serine threonine kinase that acts downstream of RAS and signaling pathways, and a relative of the EGF receptor called HER2 or ERB2. Now, about 4 years ago, 2 labs at Harvard and our group at Sloan-Kettering defined EGF receptor mutations that are associated with sensitivity to 2 tyrosine kinase inhibitors, one called gefitinib, aka Iressa, the other called erlotinib, aka Tarceva. And patients who were receiving these drugs in our clinics had very dramatic responses to these drugs, and it was subsequently discovered by sequencing a number of genes, especially the epidermal growth factor receptor gene, that the patients who developed the dramatic responses almost always had mutations that affected the kinase domain of the epidermal growth factor receptor. These mutations were idiosyncratic. About 90% of them were of one of 2 types, half mutations that changed leucine at position 858 to arginine, and deletion mutations that removed all or most of a highly conserved sequence near the ATP binding site. There are a couple of others as well that sometimes turn up in these patients who are drug sensitive. How were these mutations found? Let me just give you one vignette. In our own group, William Pao was studying the lung cancers in mice induced by mutant K-ras that I showed you early in the talk, and trying to understand this phenomenon of oncogene dependence by looking by using mice that, as I showed you earlier, lose the extensive adenocarcinomatosis in lung fields by removal of doxycycline. So when dox is removed for 7 days, the lung fields become very clear. And I show you this because Will Pao, who was an MD PhD graduate, and not only a postdoc in my lab, but also a lung oncology fellow, occasionally would see very dramatic patients in the clinic who illuminated what was going on in human lung cancer. So this woman was a woman in her mid-40s who had extensive bilateral metastatic lung cancer, as you can see by all this snowy stuff in both lung fields. She was breathing oxygen in a wheelchair 5 days after beginning treatment with one of these tyrosine kinase inhibitors, the one called Iressa. She was walking around not using oxygen and feeling a whole lot better, and you can see her lung fields are quite clear. So the relationship between oncogene dependence is demonstrated in a mouse model, and what was seen here was obviously riveting, and having individuals who can move between the clinic and the lab obviously make a big difference in this setting. So we, like our colleagues at Harvard, set off to try to understand what these mutations were about, and that's how we got involved in looking at EGF receptor mutations, and she indeed had one of those mutations. Now, despite these remarkable short-term effects in about 10% of patients, often, by the way, patients who had a history of never smoking or very limited smoking, women outnumbered men in this set, and there was a pretty high frequency of this kind of dramatic response in women from Eastern Asia. But despite these very dramatic short-term effects, the long-term effects have been less dramatic. Patients do appear to live longer when they receive these drugs for EGF receptor-dependent lung cancers, but the long-range effects are disappointing. And why is that? Well, there are probably multiple reasons, but the most dramatic is drug resistance. And we know that usually within a year, sometimes a couple of years, sometimes longer, the tumors recur, and the most obvious reason why recurrence might have occurred was based on the experience with Gleevec resistance in chronic myeloid leukemia, namely, and everyone, of course, set off to look for secondary mutations in the targets. Will Pao, for example, looked at a number of patients who had this kind of experience. Here is extensive tumorigenesis at the start of therapy with erlotinib. 4 months later, lung fields looked pretty good, but after 2 years, extensive regrowth. There was also persistent continued growth of a metastasis in the spine. These lesions were sampled, and when we looked at the DNA from these progressive lesions, we were able to show that in addition to the original deletion mutation from exon 19, there was a secondary mutation at position 790, changing a threonine to a methionine. Exactly the same kind of mutation that's observed in about 15 to 20% of patients with chronic myeloid leukemia who develop resistance to the tyrosine kinase inhibitor, Imatinib. And as this slide shows, 2 other patients, one with a point mutation at position 858, one with a deletion mutation, also have the T790M mutation. So since Will's publication a couple of years ago, there's a lot more work has been done to try to define the molecular nature of drug resistance in these patients. And we now know that his original estimate, 3 out of 6, about 50%, turns out to be pretty close. About 50% of patients who become drug resistant with either of these 2 primary EGF receptor mutations have a secondary mutation that makes the kinase inhibitors relatively ineffective in treating these tumors, and the mutation is T790M. There are a few other EGF receptor mutations that seem to confer relative resistance. I won't have time to review those. None of our patients so far have mutations in K-ras, which would be a way of doing an end run around the dependence on EGF receptor. and about 20% of patients, some concurrently having T790M mutations, some not, have been shown by Lou Cantley's lab and by Will Powell's lab, he's now an independent investigator at Sloan-Kettering, have an amplification of the gene called MET, which also encodes a protein tyrosine kinase. Now, this, of course, leaves a substantial number of cases unexplained, and it leaves open the question of how we go about testing drugs for their efficacy in the presence of resistance-conferring mutations. So one obvious question is whether drug resistance of this type, which is a major problem in trying to develop therapy for adenocarcinoma of the lung with EGF receptor mutations, that's as good as the kind of therapy that patients with CML receive, is to make mouse models. And of course, there are lots of reasons to make mouse models of this disease to try to understand progression and oncogene cooperation and so forth. But one reason we made these models was to study drug resistance. And what Katie Paletti set out to do about 3 or 4 years ago was, once we knew about the two types of mutation, the 858 mutation and the deletion mutation, was to generate what you already now fully understand. You take a mouse that expresses the tetracycline regulator, or TTA, in the lung, and you make other mice that have tetracycline operators upstream of various forms of EGFR, particularly the two forms that encode the 858 mutation and the exon 19 deletion mutation. And then you treat bitransgenic mice with doxycycline. You try to induce tumors. As you can see, regardless of which allele of EGFR is being used, these animals develop extensive tumorigenesis, seen here in an MRI image, within a few months or several months, and that the disease is, of course, confirmed histologically. The most obvious next question is whether these mutant forms of EGFR are required for tumor maintenance. Are the tumors dependent on these genes? And in this case, we can do that in two ways. We can remove doxycycline and de-induce the transgenes, or we can use drugs that are effective against the mutant receptors, namely drugs like erlotinib, aka Tarceva, to see, while keeping the gene on with doxycycline, and see again whether the tumors are oncogene dependent. So here are a couple of examples of how we do that. Here are tumors induced in the animals that express the point mutation. Here are animals that express the deletion mutation, and you can see there's extensive tumorigenesis shown in the area attached to a red T. And if we take the animals off doxycycline by imaging and by histology, it's clear that there's been massive regression. This has been documented by looking at rates of apoptosis as well. Second experiment, of course, is to treat the animals with drug, tyrosine kinase inhibitor, while maintaining them on doxycycline. Here again are animals with extensive tumors a few months after initiating treatment, induction of the oncogene with doxycycline. If we keep the animals on doxycycline and add erlotinib, the tyrosine kinase inhibitor, for a week or two, the amount of tumor mass is markedly diminished, or in this case, tumors have pretty much disappeared. In contrast, looking at animals that have tumors induced by mutant K-ras, on the model I showed you at the beginning of the talk, tumors only seem to get larger when treated with erlotinib. So given this kind of result, where we get a very dramatic response to therapy, can we begin to look at drug resistance? So we tried to recapitulate development of drug resistance by using intermittent dosing. We knew that if we kept animals on erlotinib or other tyrosine kinase inhibitors, that the tumors we didn't see any tumor recurrence. So we tried an intermittent dosing schedule. These animals remain on doxycycline, but they get erlotinib treatment 5 days a week for 4 weeks, then they have a vacation from drug. They started on drug again, another vacation, drug again, and we observed these animals by MRI to see how they're doing, and the kind of pattern we see. We won't need to go into all of this, but let's just look at the upper one. This is an animal that has the 858 mutation, huge tumor on the left. After 4 weeks of treatment with Imatinib, there's a small tumor remaining. Animals cycled on and off drug, but on drug a little bit later, 19 weeks, the tumor has clearly gotten larger. This tumor is clearly drug resistant. The same kind of thing can be seen in the other animal as well. So the analysis is fairly obvious. We sacrifice the mice when we think there's a drug-resistant tumor, harvest tumors. Most of these animals have between 2 and 5 tumors in their lung fields. In this experiment I'll describe for you, we've looked at 24 tumors from 11 animals. The first way of looking at drug resistance is to take an approach based on what we know to be the case in human lung cancers by extracting DNA and sequencing genes that are likely to be involved in drug resistance, especially EGFR, looking for the T790 mutation, K-ras, and PI3 kinase catalytic domain. And we also look to see whether any of the relevant genes might be amplified, especially the MET gene. There's also an unbiased approach we're taking, which we're in the process of pursuing, using comparative genomic hybridization or expression profiling or high-throughput sequencing to look for novel genes that might be contributory to drug resistance, and that work is simply in progress. I can't present it today. The first point to make is that in 5 of the tumors that we looked at, and I should say that Katie looked at, we found a secondary mutation that changes threonine to methionine at position 790. So the same mutation that's seen in about 50% of human lung cancers that become drug resistant. When looking at 25 Imatinib-resistant tumors, 5 proved to have the T790 mutation. 1 has a K-ras mutation, but only after about 13 months of therapy. And we know that in this strain of mice, there's a certain low incidence of K-ras mutation. None have PI3 kinase mutation. A few have a low level and amplification of some EGF receptor genes. This is still of uncertain significance. But 1 has a dramatic amplification of the MET proto-oncogene, the same proto-oncogene that's amplified in about 20% of human lung cancers. And here's just a bar graph representation of the copy number of the MET gene in a large number of tumors from untreated and treated mice. And the same animal that has an amplified gene also has high level of expression. And we assume that the amplification of this oncogene is responsible for drug resistance. But an obvious question remains unanswered: what accounts for drug resistance in the other 70% of the tumors? And we hope that the undirected approach of looking at gene copy number and gene sequence in those animals will be revealing. So just to summarize what I've told you in this story before I make some concluding remarks, it's been determined from the study of human EGF receptor mutations in human adenocarcinomas that about 10% of these tumors have EGF receptor mutations, and that these tumors respond usually quite vigorously to tyrosine kinase inhibitors, and that we can recreate the disease in animals with doxycycline-inducible transgenes, and we can also recapitulate drug resistance by using a intermittent therapeutic strategy, and we find that the same mutations that are found in human patients are also found in the mouse, encouraging us to believe that we can study the resistance and efforts to overcome it in mouse models, and also seek explanations for the other 30 or 40% of tumors in humans that don't have either of the 2 known resistance-conferring mutations. So, if there's any big summary of what I've tried to tell you today, first, mouse models appear to be useful not just for validating oncogenes and tumor suppressor genes as instruments of carcinogenesis, but also for assessing therapeutic targets and designing new therapeutic strategies, and even in genetically complex cancers, and for studying drug resistance in a manner that simulates human disease. But to make full use of all the things that need to be done in the world of oncology over the next few years, we need also to see changes that are already beginning in the culture of this area of science. I'd like to just mention a couple of those that I see happening at our place, and I think probably here as well. First is the importance of what we call multidisciplinary team efforts. So after the EGF receptor mutants were discovered in 2004, we assembled a group of people, many of whom are shown here, pathologists, oncologists, surgeons, an oncologist, another surgeon, Will Powell, who both does medical oncology and molecular biology, to form a lung cancer oncogenome group that meets once a week, has a program project grant together, has parties together to ensure that the cultures of medical treatment of cancer and the efforts to understand cancer at the molecular level are pursued in concert. And we envision even, and actually exercise even bigger team efforts. We have money from the STAR Foundation to support a consortium of investigators at our institution, Rockefeller, Weill Cornell, Cold Spring Harbor, and the Broad Institute to do collaborative research at the tune of $20 million a year to try to ensure that the scientists who have many different skills are working together to attack this problem. More globally, indeed, certainly nationally, and now fast becoming an international effort, is the generation of a cancer genome atlas, an atlas of all many of the aberrations that affect the common types of tumors that humans are heir to. This is an effort which is, again, galvanizing the efforts of oncologists, surgeons, pathologists, and molecular biologists to try to depict cancers more forcefully. The second point I'd like to make is one that I've certainly always seen at Stanford and is becoming emblematic of our place, that we have more scientists who work in both the lab and the clinic, and scientists who are trained to understand disease, even though they may not have medical degrees. And I've told you about some of these folks who sometimes have both degrees, sometimes don't, but nevertheless are sufficiently versed in the nature of human disease to focus their efforts in the laboratory on it. One way in which we're trying to promote this at our place is to create a PhD program in cancer biology, one that was somewhat influenced by my experience occasionally teaching in your own cancer biology program, but one that has a slightly different perspective because we expose our students not just to research about cancer, but also to the clinical experience of cancer. The students have clinical mentors. They spend time in both the outpatient clinic and on the wards, and they have the opportunity to see cancer as a disease that requires a deeper understanding, and understanding that hopefully reflects the needs of clinicians who have to treat such patients. This school is now entering its third year. I encourage interested undergraduates to keep us in mind. And the last item that I feel I'd always like to end with a word about sharing of what we do. We can only work as a collaborative world if we share our materials, our information, and our publications. And we as scientists need to decide whether we're going to allow our articles to be locked up under subscription, whether they're going to be made freely available through public libraries like the New York Public Library. I'm grateful to one of your colleagues, Pat Brown, for introducing me to this concept and for working with me in efforts to develop both more effective public libraries and open access publishing. That's Pat Brown. So I want to again thank the Tseng family for sitting through all this and for allowing me to speak to you today, and to many of my colleagues here for their friendship during the day, and to everybody else for allowing me to go on a little bit excessive length. Thank you very much.
So please go to the microphones with your questions so that people in the other rooms can hear.
Pat Brown.
Pat, would you go to a microphone?
So I won't take you on about mouse models, but
You can do that tomorrow.
Yeah, tomorrow. There was a particular experiment where actually I'm not sure I understood or agreed with the interpretation, which was the selectively inducing a tumor with MYC and RAS and then selectively shutting them off. And you made the argument that you could infer that RAS might be a better therapeutic target by virtue of the fact that the recurrences were slower. But inasmuch as every tumor recurred with both MYC and RAS being expressed, it seems to me that an equally reasonable model is just that the levels of expression of those two genes relative to the levels that were required just happen not to be equally matched. And in fact, the tumors were equally dependent on both of those. And a drug that effectively knocked out either one of them would have just as well suppressed the recurrence. So I'm just challenging you on that, I guess.
No, actually, I think that is one possible explanation for why, in the context of this model, it does appear that RAS is a more potent oncogene and one that's more is a more attractive target with respect to delaying recurrence. And I think you could argue that it's not inherent in the nature of MYC or RAS, but inherent in the level of expression of those genes. But that's in a sense a question of alleles. So you're digging down to another level of sophistication here. You could argue that another RAS, I'm not trying to make a blanket statement about RAS and MYC. If we, for example, had used a MYC gene that has additional mutations that confer a higher level of oncogenicity on that protein, maybe we would have gotten more bang for our buck by turning MYC off than RAS. So I think allele specificity might be an issue. I wasn't trying to say that MYC and RAS are inherently more or less important than each other. And in another tumor setting, perhaps in the lung, or which we're trying to explore, or the pancreas, or some other tissue, one is more important than the other. I just, in the case of these two alleles, that appears to be the case. And I think it is a fair measure to say that the time to recurrence is a reasonable measure that certainly the FDA likes, and I think most patients seek as a measure of success. But good comment, thank you. Paul.
Harold, in the first experiment with the MYC-RAS-driven tumors, do you see any difference in the frequency or rate of recurrence depending upon how long the genes have been turned on? So in other words, if you allow them to go with both MYC and RAS being expressed, and then you turn them both off, and then look for the rate of recurrence or frequency of recurrence, do you see any difference in how long the genes have been expressed?
Well, unfortunately, we can't really do the experiment you're suggesting because we can't wait until the tumors, well, we could do something along those lines, but we do generally wait until the tumors are about 2 centimeters in size and then do the induction. We can't allow the animals to go further than that without creating an intolerable tumor burden that makes the animals overtly sick. But we could try to de-induce the genes when the tumors are very small.
Sure.
And that's actually something we're trying to look at now in a somewhat different system where we take hematopoietic cells, turn MYC, RAS, and other oncogenes on, and ask how soon they become oncogene dependent. I think that's what your question is getting at. And there's some people who believe that there's a kind of shock effect that results from turning on an oncogene and putting signaling pathways in a kind of imbalance. And one interesting issue is how long after an oncogene is turned on, perhaps even before the cells are overtly cancerous, do they become oncogene dependent?
Homeostatic mechanisms in the cell sufficiently that they become more sensitive to regression.
Right. Another way to ask that question is, can you turn the oncogenes off more gradually and allow the cells to remain alive? That experiment's a little difficult to do because of the dosing of doxycycline, but another way to think about this problem. That's a really good way to think about it.
So I'll ask a question. It's hard, since you have animals that have many cells at risk at being clonal progenitors in the precancerous and the cancerous state. Have you looked for A, clonality of the cancers, and B, whether it's the same clone that reappears when resistance comes up? And do you have any idea if resistant pre-exists
Sure,
and
yeah.
drug selects?
So this is a question pertinent to the lung cancer experiment.
Yes.
And Irv's raising a very important question, which we're trying to wrestle with. So far, when we look at untreated tumors, we haven't seen drug resistance. But we do have at least one example of a lung cancer which never regressed in the first place. And when we look at that tumor, we see the 790M mutation. Let me say a word about that mutation. If those of you who haven't thought much about this, a secondary mutation in an oncogene has to preserve the activity of the oncogene. Otherwise, it would be an oncogene inactivating mutation. And in the case of this particular mutation, the resistance-conferring mutation is itself an oncogenic mutation. And in fact, there is at least one family that has that mutation inherited. And those members of the family that have that T790 mutation have a higher rate of lung cancer. So that mutation confers not just drug resistance, but some carcinogenicity on the EGF receptor. And it's not surprising to find that at least an occasional tumor will have the T790 mutation.
More questions? I can't believe it.
Okay.
Oh,
Yeah.
there we go.
Dean Felcher.
So, Harold, I'm curious what you're thinking now oncogene addiction really is. Because you allude to the fact that it could have to do with signaling states. You know that more recently we've speculated that it has to do with restoration of things like senescence programs or even the restoration of the microenvironmental cues like angiogenesis. I mean, clearly Judah felt that it was entirely angiogenesis.
Right, I know.
So what do you think is the mechanism?
Well, I don't think I think it's unlikely to be a microenvironment effect because you can see this phenomenon in cell culture. You take a lung cancer cell line and interfere with mutant EGF receptor, for example, or with mutant RAS by doing RNAi knockdowns, cells die. And I think there may be additional effects that are mediated through microenvironment, but I think that the primary effect, the most dramatic effect, is one that represents an imbalance in signaling pathways. So a cell death pathway turned on by the oncogene, held in balance somehow by an oncogenic effect that drives growth and has an anti-apoptotic effect, suddenly removed, and the cell death effect, which doesn't disappear as quickly as the balancing effect, drives the cell into cell death, which is an irreversible phenomenon.
I'd agree with that, but I wonder, since we're thinking about this now in a clinical context, in a cancer there'll be 100 million cells, that maybe that's responsible for 99% of what we see. But in order to completely eliminate a tumor, it really does involve considerations of things like the microenvironment, the immune system, cell intrinsic programs that have to do with stem cellness, self-renewal, and senescence.
Well, I think as I've tried to make clear, especially in the first part of the talk, I don't believe that we're able yet to cure cancers by simply exploiting oncogene dependence. There are cells left behind. The reason you see tumor recurrence even in the tumors that have both inducible RAS and MYC is contrast to the absence of tumors in animals that have the two oncogenes but never see doxycycline. It's because some tumor cells remain behind, and if they have what it takes, namely a mutation in the tetracycline regulator or in something else, they can recur. So I don't think we have solved the problem, but we have a dramatic effect. And if we can I think we face an ultimate problem in oncology if this strategy proves to be successful in a large number of common tumors. Namely, do we maintain treatment for the rest of one's natural lifespan with these very expensive, not totally non-toxic drugs, or do we try to find adjuvant therapies of some kind, whether it's immunological or something addressed to the microenvironment or multiple other kinds of strategies that we aren't yet able to predict. I don't think we yet are at the position of being able to say we've cured cancer. We cure cancer, as you know better than I, either by surgery or in a few remarkable examples like cisplatin treatment of testicular cancers, and we don't, in most cases we don't cure. And I think right now we have some remarkable new tools that give deep remissions, but until we know how to use those tools more adroitly and have other strategies for eliminating all cancer cells, especially cancer stem cells, I'll say it, then we're not going to be where we want to be for cancer patients.
Thank you for your presentation, and I have no doubt this research is going to save a lot of lives. My question follows from the last comments. Since you talked about the overlap of the clinical and the research, I'm curious what you might see arising and how soon for strategies for those groups of patients who do have the good response to the growth factor inhibitor drugs and then develop a recurrence, a resistance. Is there anything coming up that you see in the near future or
Yeah. No, I appreciate the question. I probably should have said that, for example, in the case of Gleevec-resistant chronic myeloid leukemia, there are already two other drugs, Imatinib and Imatinib, that are both very effective. In fact, maybe more effective than Imatinib itself in treating CML, and combination therapy is on the horizon. In the case of erlotinib-resistant EGF receptors, there are some other drugs, non-competitive inhibitors, that look promising but have yet to prove effective in the clinic. There are also some drugs that may work in other ways, for example, blocking the effects of chaperone proteins to try to work in combination with erlotinib. But so there's a lot being done in that area, but I didn't have a clear message to convey, so I didn't convey it. Sharon.
You spoke about the importance of open information, especially among scientists. I'd like to ask a different question about open information or communication. What do you think are the issues about communicating to the public, to the potential patients, about cancer treatment, about what works and about what doesn't work, particularly in the light of what goes on with the pricing of pharmaceuticals, pressure and TV ads and so forth about that, as well as the problems that have gone on for so long of people just thinking of completely different non-medical treatments?
Well, certainly one of the healthiest developments in doctor-patient relationships in the last 20 years has been the development of patient advocacy groups, especially those that take a very responsible attitude toward learning what science and clinical oncology are making available. And there are groups now for virtually every major and some minor cancers that try to put information into a highly accessible mode for patients and their families. There's no perfect way to do this because some people have a very high level of understanding. Someone like Alex obviously would have had a tremendously sophisticated way to understand his disease. Someone else with similar cancer might have to have things explained in much simpler terms. So I'm a little concerned about simply using the web as a way to say, okay, information's out there, go use it. I think eventually doctors and people who assist doctors, oncologists, in taking care of patients need to be able to explain in appropriate language, and sometimes a foreign language, what patients need to know. And I think one incredibly refreshing change in the field of oncology is that in part because we understand much more about the disease, it's much less frightening. You tell patients they have cancer. When I go to the clinic, it's really amazing to me to see people able to talk about their disease like it's another disease. And in a sense, sure, cancer is a terrible disease, but we have a lot of patients who have had cancer for 10 or 20 years and are working, and they understand their disease, and they know that if a drug stops working, they can try something else. So it's a disease about which we can speak its name, and while it's still a very difficult diagnosis, it's one you never get over because if you've had cancer, you're always at risk of recurrence. But the the avenues of discussion have been opened, and I think in part open because we no longer feel it's the deep mystery that it was 40 or 50 years ago.
So I'd like to ask one more question. In thinking about these really strong oncogenes like MYC and RAS, even though you've turned them on, most of the tumors you're looking at seem to be monofocal. So I'm going to assume, or make an argument, that many other events have occurred, maybe epigenetic events, in the clone that becomes the cancer cell, and then when it's under treatment, it's not starting off at the same base. It's now got like BCL2 or TERT or any of a number of things going on. Have you gone to look at the revertants to see, do they have some of these other events?
No, that's a very, very good point. The tumors that we look at actually are probably largely multifocal. And one of the things that's wrong with the TET-inducible models is that we are turning on the oncogenes in all cells. We have other ways of making models that allow the inactivation of a tumor suppressor gene in a rare cell, or the introduction with virus vectors of an oncogene into a rare cell to make tumors that really are initially monoclonal. But I agree with you, as these cells grow out, of course, the mutation rate is probably tenfold or so higher than normal. There are other things happening, epigenetic events certainly, and one of the challenges now is to try to look at what else is happening in these cells. Don't have answers for you now, but one of the things we're about to start doing, thanks to new generation sequencing, is to take some of the drug-resistant lung cancers in our mouse models and to sequence literally a few thousand genes to find out whether something other than drug resistance of the obvious kind is contributing to the growth of these cells and the recurrence of the tumors. Well, I think we all owe ourselves a little refreshment. There is, in New York we don't go out onto 68th Street, but here we can go out into the sunshine and have a glass of wine. Thank you very much.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.